Okay, welcome everybody to our monthly seminar series. It's my pleasure to introduce to you Dr. Seonghee Lee, our strawberry geneticist. Before I start my introduction, Christine asked me to remind everybody to turn off your cell phones, put them on silent. If it goes up, she will personally evict you, and we don't want that to happen.
It won't be pretty.
So, Dr. Lee grew up in South Korea, where he got his bachelor's and master's degrees in agricultural science from Seonghee University. He then moved to the US to get his PhD degree, which he got at North Dakota State University, and he did that in plant pathology, specializing in genetics of barley disease resistance. He started his academic research career in 2006. He did a postdoc at Penn State University. Then he worked at the USDA National Rice Research Center, which I believe in Oklahoma, Arkansas. Okay. And he then joined UF here in Rome in 2015, where he started in Dr. Whitaker's lab, in the strawberry breeding lab. In 2016, then, the strawberry genetics lab was introduced here, and Dr. Lee is now leading that lab. So his research focuses on strawberry genetics, and as the title here implies, his ultimate goal is to develop the perfect strawberry. I think we're already pretty close with that, but I guess we can always do better. So in his personal life, Seonghee is married to Kazuyu, which I believe works in Sam's lab. Kazuyu, are you here?
There she is, back there.
Yes. And they have a daughter, Hannah, who's 5 years old, who was born in the US while he was doing his postdocs. So in his spare time, Seonghee likes to do PCR and run gels. No, he actually does have a personal life, so he's actually got quite a personal life. He likes to go sightseeing, he likes to play tennis, gardening, riding his bike, and watching movies. So I don't know where you get all the time to do that. So Seonghee, stage is yours.
Thank you. Thank you for a very nice introduction. So thank you for everyone attending this seminar, and I'm very happy to introduce my research, and it's very see everyone in one place and talk about my research. Thank you very much. And so this is my really topic today. It's all about the genes, complex strawberry. And Dr. Vance, we take the strawberry breeder. He provided a seminar last year, searching for the perfect strawberry. Vance is all about the genes. So, but Natalia is there. Maybe she wanna say, no, it's not there. Pesticide, or acid, or the weed, entomology. There actually are a lot of things. The research actually all working together. Eventually, we can make a nice perfect strawberry. The reason the title I put here is something I want to show about my research project have been in the last 3 years, and what we are really working on the strawberry breeding program using modern advanced molecular technologies. So this is the map, the South Korea. So I was born in Cheongju. This is quite the center of South Korea. It's about 1.5 hours from Seoul, the capital. And the size of South Korea is actually really bigger than half size of Florida. And this is now the Winter Olympic is in Pyeongchang, the city. It's nearby Seoul. The Winter Olympic now, the finish actually next week. And this is actually now US map. And my science journey actually starts from Washington State University of Pullman, really west coast. Although actually I come down here Florida. So I will show how my journey started from Washington State University. So I joined the spring breeding program as a research intern for 1 year. Then the program actually started in North Dakota State University, 6th row, the body breeding program. My research was focused on the Septoria speck leaf blotch. This is one of the important fungal disease, leaf fungal disease in 6th row body. So after graduation, my PhD, my first postdoc was Penn State, and this is the only time I work on fungal genetics, not plant. So after that, I was really thought about, no, I'm the person who really love plant, not pathogen, sorry. So then I moved to the rice genetics. So blast disease is one of the most important disease in rice industry, rice breeding. So the second postdoc started in National Rice Research Center in Arkansas. Arkansas is one of the biggest rice industry in US for long grain. California is a short grain rice. So this is two different states produce 100% rice, close to 100% rice in US. So after that, 2009, I joined the Nobel Foundation. Nobel Foundation is well known for the high quality of research and Biotechnology Institute in Oklahoma. So during this time, 5 years, I spent my own research about the non-host disease resistance. What is non-host disease resistance? It's opposite to host resistance. When we look at the tomato pathogen, it affects tomato. But that tomato pathogen doesn't go to strawberry because the hosts are different. So then my research was focusing on what's going on, why pathogens know exactly the host. So my study was looking for certain genes that control non-host disease resistance. And this is my family photos. So after we moved on Florida, it's so great. Everything was weather so nice. We have very good strawberries. My daughter Hannah, she loves strawberries, picking up strawberries. And of course the fishing. So this is one time we went fishing with my friends. They got a lot of fish we got. And this is my family photo we just got a week ago. Okay. So I want to show my lab. This is a picture of my lab. My lab is really located at the corner of this center. It's hard to find. So that's the reason I show the picture of my lab. Not many people actually walk through my lab because this is really corner of the west. And this is how the lab looks like. And from 2016 October, my lab actually initiated. And so these are all our lab members from Seonghee. She's the visiting PhD student now back to South Korea. And Jimmy, she actually was an intern and she came back for PhD this year. But actually, she just graduated master's degree. Seonghee joined last year ago as a biological scientist. EPN is my technician. It's about almost 2 years now. And Hannah, she's the OPS about a little less than 1 year now. And this year I got 4 more people altogether. 3 postdocs and 1 PhD student. And Upinda, Ronald, Youngjae, and Sakya. So everyone in my lab, please stand up. I want to see it. Thank you for your hard work. And actually also many people are going to, want to know about what is really student reading program, Vance and me. So we work, we really get together, and very good communication. Sometimes too good communication. We don't talk to each other, but we have the same dress code sometimes. And so Vance is working on storage genetics and breeding. And I work with storage molecular genetics and genomics. Get together, we really work together, cultivar development. This is how we work for the cultivar perfect strawberry. And so these two the research areas that I focused on is DNA marker-assisted breeding and gene cloning and editing. So Vance has worked for trait discovery and validation. This is really important for my research. From the trait discovery, I can really move on to marker development, any other technique we can bring into strawberry breeding program. So what you see here, big map, this is all strawberry genome inside. When you look at the one tiny strawberry, it's red, it's good. But actually the inside of strawberry fruit, there's a lot of things going on. And this is a big map, and Dr. Suji Boma, last 2 years we identified all the location of traits what we are looking at, many traits. The blue circle is disease resistance, pink circle, these are all fruit qualities, many different fruit qualities. And Dr. Osorio, he's working on certain traits like powder mildew, yield, and soluble solid contents. There's many genes involved. This is not that simple. So the Louis working on special tools, statistics, prediction, many difficult mathematical calculation. He's working on that to helping the all traits get together for cultivar development. So the traits what I really focused on is simple traits controlled by single or few genes. And example, these are the flavor. We have a peach aroma flavor. This is controlled by single gene. And Young Jae is working on developing new marker to select the strawberry has peach flavor. Also, maybe some people doesn't like peach, maybe something else. And what about the grapefruit flavor? This is another flavor. Chris Bobby working on Gansby. He's a PhD student, both Dr. Kevin Porter and Vance Whitaker. And this is the one big problem in our breeding program disease, Hypothra crown rot disease. The nice thing, we have the source of resistance. And my postdoc and technician, they are looking for what is the gene that control resistance and different from susceptible cultivar. And this is another disease, Trichom crown rot. And Ashley is working. And actually, she found the genes for resistance. This is a 2 years project. She did a really good job. And how did we do? My PhD student just started from this year for this powdery mildew disease resistance. And the nice thing, again, we have very strong resistance germplasm for powdery mildew. Now we are looking for genes, what is really going on there, to identify genes and bring into new cultivar development. And bacteria angular spot. And what you see, the tiny specks, the brown spot, that's all the disease symptoms, get the tissue to dead. And when you look at it upside down, the leaf tissue, there's a wet, softened. This is very typical symptom for this bacterial disease. And nice thing, we also have the germplasm for resistance, and my PhD student, Jin Yi, is looking for genes which control this resistance. Anthracnose, Natalia Salinas, is PhD student and Dr. Vance Whitaker, and somehow we have all disease resistant source. This is really power of our breeding program. We can find things in our germplasm. And that genes, Natalia found 2 genes, I believe, or one major gene that control from here to here. So this is a very nice thing. And another disease resistance, charcoal rot. This is how the plant look like, the gut disease. And John Nelson is working on his master degree program to identify gene for the resistance. This is a resistant genotype. And so we have a very nice traits that controlled by single or few genes. So from that point, we can publish very nice paper, but that's not the end for breeding program. We have to bring into the application and develop a new cultivar. We have to plant in outside, in the field, right? And so this is what the technique, the method we are using, we call molecular assisted breeding. For this program, what we need a DNA marker. And using DNA marker, we can do marker assisted seedling selection. And using that all together, we can develop cultivar for disease resistance. So what is the DNA marker? I want to just explain a little bit about the DNA marker. What you see in red and some kind of line, these are the DNA in strawberry. So these are the DNA is double-strand DNA. And you have many genes connected together. Actually, you make a whole genes, all the DNA. And let's say one gene. So every gene is actually represent unique phenotype. Let's say one gene belongs to white strawberry. DNA, the genetic code, very simple, just a 4 possibility, A, T, G, C. Every living organism is exactly the same. Strawberry, insect, human body, everything same. Whatever A, T, G, C, it shows all the combination, possibility that make a combination that change all the phenotypes, from eye color, hair color, strawberry color, everything. So when you look at here closely, there are some sequence, they are different. They are different. The different means they will change the phenotype. Red strawberry to white strawberry. So for that, so the two flag is a DNA marker. We can target the unique DNA sequence change as a DNA marker. So DNA marker can detect any seedling, any genotype what we want for the phenotype. For this case, is white strawberry. This is one simple example how DNA marker can be used. When we use GD is a gamma, GD is a peachy aroma marker. Okay. Also, the white strawberry can be another marker. For this seedling, you don't have fruit. How you can phenotype? You don't have any idea. Using DNA marker, we can select because this marker is unique. They can detect any seedling has a peachy aroma gene. From there, we can choose all the seedling which one has this gene, and we can bring into the field. This is 100% selection into the field. This is the power of DNA marker. We can really select seedling, bring into the field, and we can test for our breeding program. Gamma D galactone is the peach aroma, tiny, tiny molecule. This tiny, tiny molecule, sweet sensation. This is one of our sweetest variety. As gamma D galactone, this tiny molecule, and has a nice, good flavor, peach aroma. And Dr. Alan Chamber, he identified this gene, FAD1 gene. He's the now faculty at Homestead. He developed a marker for this gel-based DNA marker. It works very well. You can detect everything, this DNA fragment here. These are all, you can say, peach aroma prediction. It's marker very well. And when we screen many samples, this is how the conventional DNA marker, gel-based marker look like. You have to learn many agarogel and screening, taking picture. It takes forever. It works well for small sample, but what about a large scale sample population? Our breeding program, we cannot deal with a few samples. We have many samples. So this is a high performance large scale genotyping we introduced last year. This is the automatic machine, you can get genotype. To use the genotype using this machine, very basic marker, it doesn't work. We need a high throughput marker, different level of marker. I can explain a little bit about how I explained about what is DNA marker. Now I can explain how we develop DNA marker. To understand that, I want to explain a little bit about the genome strawberry. What you see here, this is tiny strawberry, white strawberry. So woodland white strawberry, first genome fully sequenced a few years ago. There are 14 chromosomes. 2 chromosomes get together, one unique set of chromosome set. So that means 7 sets of chromosomes. Each set chromosome, they have unique characteristics. So what we call this is a diploid, because 2 chromosomes get together as a one unit, so it's a diploid. What about the cultivated strawberries? We also have exactly the same 7 sets of chromosomes. That's it? No, we have another copy. We have another copy. We have another copy. What that means, this is lots of things going on. And when you look in here, the 7, they are similar to each other. These are all one group, another group. But they are similar, but they are not exactly the same. And that's what we call 2 multiplied 4 is 8. So it's octogroup. We call octogroup. And this is already many people should know about the diploid small strawberry genome getting bigger. We got big strawberry. This is how they actually genome contribute to larger fruit size. Who did that? This is actually what breeder did. Breeder, they artificially cause. They make a genome larger and larger and larger. Eventually, while we're using octogroup, this is the best commercial fruit. And this is the naming system we made. 1, 2, 3, 4, 5, 6, 7. And from this line, A, B, C, D. So then, for our breeding community, or strawberry community, everyone knows when they say 7A, they know exactly 7A. 5C, 5C. So these the name represent unique genetic characters. So I can explain how we develop marker for gamma D-galactone, Fitch aroma deselection. So FAD1 gene located in 3B. A Winterdome doesn't produce gamma D-galactone. And sweet cherry, this is one of our parent we use for this gene identification. Sweet cherry, probably many people know here when we have tried the sweet cherry, it's sweet and has better flavor than Winterdome because of this gene contribute that. What you see is a 9 here, individual chromosome 3, A, B, C, D. This is when we look here, each chromosome set, this is from starting to end DNA sequence. So when you're taking out, this is kind of the line from the thread line. We have a tiny, it's just a line, keep going. This is exactly the same thing in the chromosome. So you can sequence from end to start to the end, get all sequencing code in one line. From there, we can identify where is the location of this gene. This gene actually is in 3B, is supposed to be here. And let's just zoom up a little bit, zoom in there. And what you see here, tiny different colors, small, this bar. This is an old piece of DNA sequence product. How we do DNA sequence? This is one chromosome. And make a small, tiny fragmentation. We can just cut many millions of small fragments. Then using DNA sequencer machine, we can just sequence. We can read every this small fragment. After that, computer, software, bioinformatics, every tool, we can align all together. So then when the sequence they similar each other, they can get making group together. Eventually, it's making like this, what we call this genome assembly. And winter-dawn, clearly the gene is missing. Sweet Charlie, the gene is there. So this is how we found the gene using DNA sequence techniques. And using this gene information, we can develop DNA marker for gamma-D galactose, the peach aroma. So now we have a marker. So we can use that for many seedlings for our breeding program. So to test the DNA marker first, don't go to too many, everything to big population. We have to make sure this marker works for our all parents, for our cultivars. Radiance, sweet sensation, fruit of beauty, brilliance, sweet Charlie, alien, and winter star. We already know these are all producers. And the marker, 100% show very accurate. Other California varieties, Monterey, Portura, Canarosa, and the other festival, we just done our two Florida variety doesn't have gamma-D galactose. This marker show absence. This is 100% match. From there, we have a marker. Let's use our marker for our real breeding program. Large, many samples. What we normally do, we do 40,000 seedlings screening breeding season. It has to be done 5 weeks to send all our seedlings to California nursery for next season our breeding program. So this procedure, what we call marker versus seedling selection, mass. These are all seedlings we have to test. Using this technique, we don't have a really time for classical DNA extraction. This is one tiny deep disc punch. You can buy in Home Depot or Staple, the tiny punch. Then put in the DNA plate, the PCR plate, and get 10-minute DNA extraction. We call rapid DNA extraction. Get all this genotyping, and we can just do computer analysis. This is the reason how they look like. This is all blue lines. Many seedlings actually that result overlap. And this what peak that means has the gamma-D galactose, the cultivar, the seedlings, any genotype, the plant materials what we're using it has the gene. So we select that only seedling, we send to the field. Whatever the other doesn't have, we can toss out. We don't have to maintain the seedling because of the land charge. Every procedure is too much. I want to show one plate. And this is the one that genotyping plate. 384 samples, 1 hour, you can finish. You can finish 1 hour, 384 samples. And how much the cost is? This plate is $2. But the total cost, everything except the labor, 10 cents per sample. It's very good, in-house. If we send out, it's very, we cannot handle that. It's also the time we cannot handle. So everything is critical. We have to do everything in our breeding program. So this year, so when I joined in 2015, I worked only one trait. This year we have many traits. We have to select. These are peach flavor, Phytophthora crown rot, bacteria angularis spot, corrected trichome crown rot, anthracnose, and day neutrality. These all markers we already have. This is ready for marker assisted selection. The power of this high throughput mass is we start initiate 40,000 seedlings, we go down, finally we just need 10,000 seedlings. This is the size of population we need for the field experiment. So these are all 10,000 seedlings. We can send everything for our field experiment. So what they call this 100% selection by markers. So next topic I want to explain about gene cloning and editing. This is a more advanced technology what we recently introduced in our breeding program. Florida Festival has the gene for Phytophthora crown rot resistance. This is advanced selection line, the number doesn't have the gene. So one single gene changes the life of strawberry plant. Healthy, very good, looking good. This is completely just dry out there. This is controlled by one single gene, PC2, we call PC2. Where the PC2? PC2 is 7D here. So Eliana, this is also has PC2 resistance. Sweet sensation. Sweet sensation has very nice fruit quality. One of the problems is doesn't have a resistant gene for Phytophthora. So that's what we're working on, to bring in the Phytophthora resistant gene PC2 into our new cultivar. So this is a chromosome sequence view, 7A, 7B, 7C, and 7D. This gene located here. This is actually power of the sequence. When you have a sequence, full genome sequence, this looks like just picture and a lot of things going on complex. But when we have a sequence exactly each chromosome, we can identify the gene exactly where it is. The location is very important to develop a marker for the research. My postdoc, bioinformatics, genomics analysis postdoc, is the sequence all resistant cultivar, Canarosa, Eliana, all other advanced selections. And also sequence susceptible cultivar or other genotype, advanced selections. The sweet sensation, brilliance, the others. These are the sequencing methods what we use. This is a piece of DNA. As I mentioned earlier, DNA code is photon ATGC. And this machine can going down this direction. It's like chain, and giving all the sequence information coming out. So we can make one nice sequence result from here. What is really different between R and S? What DNA really changes the life of the plant's dead and alive? These are the DNA marker we use for Phytophthora PC2 collection. We identify when we're looking at the gene getting inside of the location, we found identify exactly the gene. These are the disease-resistant gene. And this gene already has been published in soybean Phytophthora disease resistance. That means it's a highly possible this gene works for the strawberry Phytophthora disease resistance. This is one thing interesting. This is one of the preliminary data, which I want to show. The Eliana sweet sensation, what the chance of difference is really tiny. We found that there are small changes of DNA sequence. It changes between resistant and susceptible. So now we have a clear idea what kind of gene really control Phytophthora disease resistance. Then the other topic we can read now, we have a DNA marker, already good quality. We know the gene. Another technology what we can use gene editing. Many people should have heard already CRISPR-Cas9 gene editing. This is the breakthrough technology what breeding program or human or animal, a lot of people actually moving on this research. Previous slide said, I showed just tiny changes between resistant and susceptible. And these 4 DNA code between susceptible and resistant, we can just edit. This is one technique. And these are the sequence we can actually use and change this method. Gene editing is non-GMO. Because why? Everything is happening in strawberry genome. We are not putting any foreign DNA. Everything, we know exactly what's going on. The sequence, the small piece of sequence are changed in susceptible. That's why it becomes susceptible. We can just change the sequence. This is what we call gene seizure. We can cut a special enzyme, we can get in together. Then we can make a resistant cultivar. This is one technique. To use gene editing, this technology, we cannot just use this regular plant. We have to go through everything, tissue culture system. So we are establishing this tissue culture system for gene editing, two main cultivars, Florida sweet sensation and brilliance. So my biological scientist Chun Min is testing which part of strawberry tissue we can use for best for tissue culture. This is pegiol and stolon and other all different parts of stolon piece. And this is all the plants image where you see the all different selection, the media, hormone. This is a lot of work. So this picture shows we optimize all tissue culture condition for sweet sensation. This is the one cut from the run-up tip. Then you can go to generation in the media, and you see actually this is what the shooting strawberry. Shooting is from the petiole or run-up tips. And after that, 2 weeks later, you have a whole one nice single plant and root got regenerated plant. This whole procedure normally takes 3 to 4 months. And this is the same example in Florida brilliance. So these two cultivars, we have to use different tissue culture system. They look like same strawberry, but tissue culture, the hormone, the nutrient, everything, these two cultivars requires it quite differently. So I want to explain gene editing procedure. This is a very simplified gene editing procedure. From here to whole plant, which step we have to use for gene editing. Gene editing, we need to deliver our special edited gene construct in this system. So this is kind of the picture where we can make a special gene construct, change small DNA sequence, which change is resistant to susceptible or susceptible to resistance. Any direction of change, we can make plant resistance or resistant plant becomes susceptible. They change the gene, like this method. We are not using any bacteria for transformation. We're using method from here to there. This is the tissue stage. We use a special method using gene gun. Gene gun is this is the machine. Actually, this is the part of gun. You can shoot the gene into the tissue culture. For example, this change of DNA sequence, we can actually coat gold particle. We need a kind of something special metal or round things, very, very tiny. This is, I believe, a 4 micrometer or 10 micrometer. So that small particle we coat with the DNA. So gold particle we shoot. It's a gold bullet, tiny bullet. It can go directly into the plant tissue. Once the gold particle get landed inside the plant tissue, the DNA release out. They can go inside of cell. So how this way we can actually target strawberry cell, and this is one of the tests how our gene delivery system works. We use a GFP protein, green fluorescence protein. This protein initially isolated from jellyfish. Using this GFP protein, we can use gene gun and shot into strawberry tissue, the embryogenic cells, that we can use in turn off the light. We can use microscope. There's the fluorescence light you can see here, many. One single fluorescence, that means the GFP, the protein is expressing there. So the delivery system works. So So these are some of the traits also we're gonna use for gene editing. So now my postdoc is working to identify the gene for fruit color. So we have white strawberry, and this is the cultivar we are looking for developing next few more years. And these are the population actually we have. When you're looking here, red to white, clearly it has segregation. That means there's genetics involved. With certain genes, when we cross parent A and B, and this naturally they are selecting, they are segregating. So that means there's a genes involved color red to white. We want to clone, we use as a gene editing technique. PC2, this is the one I already explained there. And XF1, this is the gene we found control angular leaf spot. Only one bacterial pathogen in strawberry. This is a single gene control susceptible to resistance. So we want to clone this gene and use also as a gene editing. This is one of the early stages of gene editing. So in the future, this technique will get more that will used in the public or the research institute, and this can be very powerful method for future strawberry breeding program. So one thing that my conclusion slide, the perfect strawberry you cannot make only one person. Everything is really great teamwork. I really thank everyone in our breeding program, Vance, and my program. We're really working together as a one team. We are not single individual researchers. We are really working together one team, making perfect strawberry. I think this is a really conclusion result for our future. The one I want to acknowledge very important, the support for my research. FS, REF, FSJ, Kenneth Parker, thank you very much for supporting our breeding program. Without years of really hard to get very nice results. Recently got the USDA SGRI grant, and also I involved the Rose Breed. These are the all different institutes. The small fruit breeders, they get together. This is one of USDA support to the funding. And also IFAS, a lot of support from my program. And also GCREC, this is the best support of my program. Thank you very much.
Thank you, Seonghee. Now we have some time for questions. Any questions for Dr. Lee?
Thank you, Dr. Lee, for breaking it down so I could actually understand it. You did a fantastic job.
Thank you.
Thank you. Have you put any thought into what you're going to do with Dr. Perez when you solve all the pathological problems? Are you going to have a place for her in your breeding program?
That's a good question. But the one thing, pathogen, they are not that simple. The Natalia already knows. Pathogen keep doing evolution. Once we use the gene, right, the pathogen, they know how to survive. That's why Phytophthora, think about how many years in the storage field, they never be enough.
I think I'm saying it's the nice pathologists that won't have a position. But I do have a question too about the flavor, because I know about the business part. But are we only selecting for the peach flavor, so if the seedling doesn't have the peach flavor, it's thrown away? Or because I see flavor is very complex, so people might like the peach flavor a lot of tone, and are we discarding everything that doesn't have the peach flavor?
Right. Our main goal to use marker for peach, for diseases is we want to try maximum possibilities taking the depth. All trait what we're looking for is a unique. If peach aroma gene doesn't have the seedling, it's possible you still have some other volatile matter, other flavor there, but there's going to be other pathway, not the pathway through the peach. And But the problem, the size of our breeding, 10,000 seedlings, we need to put in there. If you have more land, more ability, we can increase more, we can open more. This is a very kind of the normal. Is it right, Vince?
Well, I think the key concept to remember is that we're testing a fixed number in the field. So, thank you, Joe. We're testing a fixed number of seedlings in the field. What we don't do is say, okay, I've got 100 seedlings in this cross, 50 of them don't have this, and so they're gone, and I test 50 less seedlings. No, we start with 1,000 seedlings in a family and cull them down to the 100 we want. So we're not using the absence of the gene to get rid of otherwise good strawberries. We're just stacking the deck for what we want in the ones that we do test. So we're not testing fewer seedlings because of this. We're starting with more and testing the same amount. Does that help answer your question?
Yeah, it does.
Okay.
Thank you.
Any other questions?
Dr. Lee, do you think is it possible with these new techniques to have the perfect strawberry with perfect flavor and with resistance to all the diseases that we have?
Yeah.
In a same cultivar.
I think so, but the question is how long it's gonna be take? How many years? Right. That's what we go into that direction. Right.
Well, you still got some use.
Dr. Lee, when are you going to test the peanut flavor with the strawberry?
Peanut flavor?
Yeah. Peanut.
Oh, I don't think we select peanut flavor. Oh, you like it? Okay, then that's the rest of the peanut. But I didn't mention one of the caramel flavor. We actually thinking about to select caramel flavor. We actually putting more, little more chocolate flavor. Yes.
Are you testing any susceptible genes like MLO for powdery mildew resistance?
Yes, that's the Ronald. He's planning to work on that. So the MRO is the recessive gene. If the MRO has multiple genes, all is a gene family, then all the gene going out, then become resistance. That's what Ronald is planning to work on that too.
Do you know how many copies do you have in strawberry?
I heard that it's not more than 20. I know that around 15 should be. Yeah, exactly, I don't know. But it's around 15 to before not more than 20. So cultivated strawberry total gene number is 200,000. Then we are looking for one gene out of 200. This is a lot of work. It's not that simple, but we have a really great team. They're working all together as a one single team. That's why everything is really possible. What we've shown here, all the traits, gene, everything has been done last 2 years. This is really amazing how much we achieved recently.
Anybody else? All right, well, let's give Seonghee another hand. I just have to make one brief announcement for our next seminar, which is going to be on March 30th, and it's going to be Dr. Kozer, Andrew Kozer.